Hey everyone, welcome back to my channel, and today I'll be sharing with you a pharmacology tutorial about antibiotics and antimicrobial drugs. First of all, let me give you a very brief overview about the structure of gram-positive and gram-negative bacteria. I hope you remember that the bacterial cell wall is made up of peptidoglycan, and the gram-positive cell wall is thicker in contrast to gram-negative cell wall, which is thinner, and both of them definitely are made up of peptidoglycan. Notice that in finger number 1 you have a thick cell wall. This shows gram-positive bacteria. This thicker cell wall actually retains the primary dye, which actually stains at violet or purple. And notice there is addition of an outer membrane which binds to safranin in case of gram-negative bacteria. And also you can see a small structure called porin in the outer membrane of gram-negative bacteria, and it is a structure which allows the entry of different substances inside the bacteria. Now let's take a look at how the synthesis of cell wall in case of bacteria occurs. Since peptidoglycan are the monomers, are the building blocks for cell wall synthesis, I have shown over here the two steps which are required in the synthesis of cell wall. Number 1 is transglycosidation, and number 2 is transpeptidation. Transglycosidation actually is a work of transglycosidases, a group of enzymes that joins the peptidoglycan monomers together and in chain monomers together. And the second step, which is transpeptidation, is a work of transpeptidases, and they actually cross link the chains of peptidoglycan, and that's how this simple process of cell wall synthesis occurs in 2 steps. Now, having said all of this, number 1, I want to tell you about vancomycin, which actually inhibits transglycosidation. Since this is a tutorial of pharmacology, we'll be dealing with mechanism of action of different drugs. Vancomycin actually inhibits this first step, transglycosidation, by inhibiting transglycosidases. The penicillin, on the other hand, actually inhibits this cross-linking, this transpeptidation, and actually weakens the cell wall and kills the bacteria. So now let's properly start our topic. Actually, there are different subclasses of antimicrobial drugs or antibiotics. Number 1 is beta-lactam drugs, and they include penicillin, cephalosporin, carbapenem. And what are beta-lactam drugs? As their name indicates, these are the drugs with the structures containing a beta-lactam ring. I have drawn for you penicillin and cephalosporin structure, so you can see there is a ring which is a beta-lactam ring, and thus they are named according to their structure. Please remember that this beta-lactam ring structure is actually needed to be intact in our antibiotics for their efficient work, or for their efficiency, for their potency. This structure needs to be intact for the antibiotic to function. With this, I want to clear 1 to 2 terms. You might have heard about beta-lactamases. Beta-lactamases are actually bacterial enzymes. You can also call them penicillinases or cephalosporinases. Ases means enzyme, something that is acting as enzyme. Beta-lactamases are actually bacterial enzymes that hydrolyze the beta-lactam ring structure of penicillin and cephalosporin. So the ring structure which was present in the structure of penicillin and cephalosporin, that is hydrolyzed by any bacteria that produces beta-lactamases actually, and thus it confers resistance. So it is one of the mechanism of resistance. Another term is penicillin binding proteins, and these are a group of specific enzymes which are actually located in the membrane, cytoplasmic membrane of bacteria. Penicillin binding proteins are those enzymes or those proteins to which our penicillin will bind, and it will carry on with its action. So one more time, beta-lactamases are those bacterial enzymes which have the ability to hydrolyze the potent ring structure in our penicillins and cephalosporin, and thus they are a mechanism of resistance. And one more term I want to add, beta-lactam inhibitors. Beta-lactam inhibitors are the inhibitors of beta-lactamases, meaning this is something that will protect the hydrolyzable penicillins from inactivation. Means our penicillin won't be inactivated if they are attacked by beta-lactamases, because beta-lactam inhibitors would be present and they will be inhibiting beta-lactamases. The example of such beta-lactamases inhibitors are clavulanic acid, sulbactam, tazobactam. Now, with all of this basic overview in your mind, I hope you'll be able to pick up the mechanism of action in different terms that will be coming along your way. So, number one, in the beta-lactam drug, the drug which I'm going to discuss is penicillin. Penicillin is actually a bactericidal drug, that is, it eradicates infection by killing the bacteria. And about mechanism of action, penicillin actually binds to penicillin binding proteins, and it inhibits the step which is transpeptidation. I have told about this previously. It inhibits transpeptidation, and thus cross-linking does not occur and cell wall weakens. Now let's move towards the subgroups of penicillin, and we'll be covering the uses alongside. Number one, the subgroup is very narrow spectrum, and it includes methicillin, neficillin, and oxacillin, and it is used against Staph aureus infections. And unfortunately, we also have MRSA. MRSA is something which is methicillin resistant Staph aureus. It is Staph aureus which has produced resistance against methicillin, and the way it does it is that it produces a structural change in penicillin binding proteins, and due to which methicillin cannot bind to such PBPs or penicillin binding proteins, and resistance is produced. And if your Staph aureus is unfortunately MRSA or methicillin resistant, then you cannot use methicillin in it. But if it is methicillin sensitive, you will use methicillin. This group also contains antibiotics, usually which are beta-lactamases resistant. The second group of penicillin which you see is narrow spectrum. It includes penicillin G and penicillin V. The narrow spectrum penicillin group includes penicillin G and penicillin V, and penicillin G is used for syphilis. Benzathine penicillin G is a very good drug of choice for syphilis, and penicillin V is usually used for oropharyngeal infections. And the organism which it can cover is streptococci, usually pneumococci, meningococci, and syphilis definitely, so treponema pallidum. This is very important to remember, so please try to remember it. So basically it covers gram-positive group. Narrow spectrum usually covers gram-positive. The third group which we'll see is broad spectrum, which includes ampicillin, amoxicillin. It has broader spectrum because it is being used against some gram-negative bacteria as well, and gram-positive alongside, and it can be used against E. coli, Haemophilus influenza, Listeria. Listeria is important to remember, Enterococci, and all of them use ampicillin. And there are different infections of like otitis media, Borrelia pylori. Amoxicillins can be used for otitis media especially, so you need to remember this. The last subgroup is extended spectrum, or you can say very broad spectrum, and it includes piperacillin, ticaracillin, and it is used against Pseudomonas, Enterobacter, Klebsiella. And the way you can differentiate it from broad spectrum is that broad spectrum covers gram-positive as well as some gram-negative coverage as well, but extended spectrum gives you a very extended spectrum of coverage for gram-negative, a very vast, many gram-negative organisms can be covered in case of extended spectrum, a very wide range of gram-negative bacteria. So the coverage against gram-negative actually increases down this chart. The first very narrow spectrum was only acting against Staph aureus. The second narrow spectrum was giving streptococci coverage and some gram-positive coverage, but no gram-negative, and it has treatment for treponema pallidum as well. And third group actually gave a few gram-negative and almost all of the gram-positive bacteria. The fourth one is giving a very extended coverage for gram-negative and definitely all of the gram-positive as well. I have just tried to cover the subgroups and uses precisely. I hope you get it. I hope you pick it, and I hope it helps. Now, a few words regarding the elimination of penicillins. It occurs via active tubular secretion, a secretion occurring in the PCT of the kidneys. And there is an important point to remember, which is secretion is blocked by probenecid. Probenecid is a drug of choice for gout. So, if a person has gouty arthritis, he or she is taking probenecid, the level of penicillin will remain high for a longer period of time in his body because secretion will be blocked by probenecid of penicillins. And another point to remember is naphthalene, oxacillin is excreted in the bile. The rest of the penicillins are excreted via kidney by active tubular secretion in the PCT. The side effects, like similar to other antibiotics, they can be nausea, vomiting, diarrhea, and they may range from a minor rash to anaphylaxis. And you have to assume complete cross allergenicity between the individual penicillins. And there is a special term which is Jarisch Herxheimer reaction, and that's what you encounter in after a few hours of the treatment of syphilis. There is a reaction of the body to dying organisms which occurs like fever, muscle pains, and that's actually the reaction to the dying organisms of our by our body. That is given this special name, and it occurs in the treatment of syphilis, one of the side effects of penicillins. Now I hope that completes some basic discussion regarding penicillins. I'll see you in the next video with cephalosporins.